about the clinical research. So first we need to understand what is clinical research. So there might be many definitions of clinical research, but the definition which defines the clinical research clearly is that is a study of health and illness in the people. You study the health, like why people are healthy, what are the parameters which are responsible to keep one's body healthy, and what are the parameters which are responsible to keep one's body unhealthy. And once the patient is suffering from a disease, then what are the things which can be done uh, to get rid of that particular illness. So clinical research in particular is the study of health and illness in people. Okay, so it's a way of research to learn how to prevent, diagnose, and treat illness. Okay, now clinical research is a part of healthcare science. Healthcare science is a big umbrella, and clinical research is a part of that big umbrella, which determines the safety and effectiveness of the medication, means drugs, the medical devices such as pacemaker, etc., or any diagnostic product and surgical procedures. Okay, so in clinical research, we determine the safety and effectiveness of medication, medical devices, any diagnostic products, and any surgical procedures or operations. Okay, so now what is the difference between clinical research and clinical practice? So clinical practice, like whenever you are visiting a doctor, what he is doing clinical practice, because he is already writing the products which are already approved, which are already well tested. Okay, while in clinical research, what we do, we gather the evidence. Why we gather the evidence? We gather the evidence to establish safety and effectiveness of drug product, so that it can be used in clinical practice. Okay, so in clinical research, we are going to generate the data. We are going to generate the data, the evidence, so that we can establish the safety and effectiveness of the drug product, or medical devices, or surgical operations, anything. Okay, while in clinical practice, we already deal with the approved product, for which we have already established the safety and efficacy. Okay, now the question comes. In previous slide, we learned that in clinical research, we are going to establish the safety and effectiveness of medication, devices, diagnostic product, and surgical procedures. Now the question comes, how we do that? Like how we determine the safety and effectiveness? Okay, so to determine the safety and effectiveness, what we do, we do the experiments. Now those experiments which are done to determine safety and effectiveness are called clinical trials. Okay, so clinical trials are nothing but experiment which are done on the human beings to determine safety and effectiveness of drug product, medical devices, procedure, or any diagnostic product. So clinical trial always are designed to answer a specific question to determine whether new drug device or procedure is safe and effective or not. Why specific questions? Because every every experiment will have some objective, right? If you are doing any experiment in a lab, you will have some objective. Similarly, every clinical trial will have some objective. So every clinical trial is designed to answer a specific question, to answer that specific objective, to determine whether that new particular drug, device, or procedure is safe or effective or not. So clinical trial is a part of long drug discovery process. Okay. So it's an experiment. It's a part of a very lengthy drug discovery process that takes many years to complete. A molecule from the lab to the market will take at least 15 years. Okay. So now we learn in that long drug discovery process where the clinical trial lies. Okay, so the experiment of a drug or a medical device does not start from human beings. Okay, before starting any clinical trial, we need to generate some data to determine that drug is really effective and it's really safe. So now we learn the drug development stages. Okay. See, before clinical stage, as I said, that in clinical stage we are dealing with a human being. We will give the drug to the humans. Okay. But to give the drug to humans, it's a big thing. We have to deal with number of things. We have to see the ethical part. We have to see whether the drug is really safe or not. So to start the clinical part of a trial, there are two initial stages, that is discovery and preclinical. Okay.
Hello, sir, you are not audible. Please unmute yourself.
So since how long I was not audible?
Just now only.
Just now only, right?
Just now, sir.
Okay,
Just
okay.
now.
Before that, till this slide, you were there with me, right?
Yes, sir. Yes, sir.
Okay, okay. I'm sorry for that. It's maybe my mistake. And thank you for letting me know this. So discovery stage of development itself is having three stages. That is target discovery, target validation, lead generation, and refinement. Now we will see what does it mean. So target discovery. So in target discovery, what do? We will identify the protein, gene, receptor, which are responsible for that particular disease. Okay. For example, somebody is suffering from hypertension. Say like 50 years back, we were not sure why the people suffer from the hypertension or high blood pressure. We don't know. So in a target discovery, what do? We try to identify the cause of that particular disease. Okay, then only we can create molecules and we can develop the drug, right, in later stage of the development. But initially we need to understand what are the different reasons to have which are causing that particular disease. So we will identify the proteins responsible for the disease, gene responsible for the disease, any receptors which are responsible for that particular disease. So always the sponsor companies or pharmaceutical companies want to target which meets its clinical and commercial requirement. Okay, so it's a big subject. For any sponsor companies or any pharmaceutical companies, they will invest on a disease uh, which are related to the mass, right? That's why the sponsor of pharmaceutical companies are reluctant. They are hesitant to invest the money on rare diseases. Okay, because they have to see the commercial part also. So in target discovery, any pharmaceutical companies will also see the commercial requirement. Okay, before investing in any drug research. But in general, but in general, if the research is being done in an institute or in government bodies, then they will not take care about this part. But they have to identify the protein, gene, or receptor which are responsible for that particular disease. Now the second question comes: target validation. We got some clue in the target discovery stage. Okay, this particular protein or these particular proteins are responsible for that particular disease. For example, protein A, protein B, protein C are responsible for blood pressure. So now in the target validation stage, what we will do? We will create animal and cellular models. Okay, by doing different genetic manipulation, and we will see whether that particular disease is appearing in those genetically modified model or not. For example, in our previous example, we were studying the blood pressure. So what we will do? We will modify that particular protein or receptor which we found out in target discovery. We will modify those particular gene or protein in animal models, and we will see whether after modifying those particular gene or protein, whether that animal models are going to have that particular disease or not. Only then we can confirm, right? Because we have modified that particular gene or protein. That's why this particular animal is having this particular disease. So in target validation, it's kind of confirmation of our initial discovery. Okay. Now, now the third stage comes, that is lead generation and refinement. Okay. So in lead generation and refinement, we will create number of molecules. Okay, that is called hit discovery process. We will create number of molecules and we will perform the screening. We will develop many methods to screen those particular molecules. Okay, nowadays in labs, for example, in CSIR lab or ICMR labs, we have robotic process that is called high throughput screening. So in that robotic process, number of molecules, thousands of molecules can be screened altogether. When I say screen means whenever they develop some molecules, they will check the group whether this by adding the chloride group or by adding the methyl group, if the characteristic of that molecule is changing or not. So they will do the design of that molecule. They will check the molecular weight, etc. Okay, so they will refine that particular molecule and they will come up with one molecule which is having some good activity against that particular disease. Okay, that we have established in the target validation stage. Okay, so in target discovery, we come up with the reasons why that particular disease is happening. In target validation, we have confirmed, okay, these are the regions which are responsible for that particular disease. In lead generation and refinement, we have created molecules for that disease. Okay, now we have number of molecules that we have created in the discovery stage of development. Now we have to test those molecules in animal models again. So once we have a lead molecule, once we have a molecule which is the chemistry of that molecule is good, right? The molecular weight is optimum. It's having uh, it can be absorbed easily by the body, etc. So once the desired character, the molecule which is having the desired characteristic is identified, we will we will test that molecule in animal models. Okay, so in preclinical stage of development, what we are going to do, we are going to we are going to run an experiment with a lead molecule in animal models. Okay, we will have rats, rabbits, and all, and we will check. What we will check? We will check the efficacy, toxicity, and pharmacokinetic information. Now the question comes: what is efficacy? Efficacy means how successful is the performance of drug candidate? For example, shrinking of tumor by anti-tumor drug. For example, if we are doing research on anti-tumor drug, in that case, whether our drug is able to shrink the tumor size or not, that would be its efficacy. Okay, efficacy means how well the drug is performing. Then, what is toxicity? Toxicity means whether it's doing any organ degeneration or not, whether rats are dying because of this drug or not. So those kind of things we'll check in toxicity. Okay, uh, toxicity may be reproductive in nature. For example, rat may not have that toxicity, but it will have some toxicity for it will cause some toxicity for embryos. Okay, so those kind of things we will check in toxicity parameters. Now, third thing is pharmacokinetic information. So, what is pharmacokinetic? Pharmacokinetic means how well the drug is getting absorbed in rat body or rabbit body or any other animal that you are taking for preclinical stage. How well it is distributed in that animal body, how well the drug is getting metabolized in the body, and how it is going to be excreted through urine, through saliva, through tears. Okay, so these are the things we will check in preclinical stage of development. So the key takeaway is we will check the lead molecule in animal model for efficacy, for toxicity, and for PK parameters. Okay. Now the preclinical stage of development follows certain guidelines. Okay, they follow GLP, good laboratory practice guidelines they follow, and the quality and integrity of non-clinical laboratory studies are monitored by GLP guidelines. Okay, now in discovery stage of development, we came up with some molecules. In preclinical stage of development, uh, we have done the experiment with the lead molecule to determine the efficacy, toxicity, and PK parameter. If the results are fine, means the drug is not much toxic, it's efficacious, and PK parameter is also good. In that case, with that lead molecule, we'll start the clinical stage of development. Okay, so the clinical stage of development is also further divided into four parts: phase 1, phase 2, phase 3, and phase 4. Now, what we do in phase 1? So, first of all, phase 1 is a first in human trial. Okay, because till before phase 1, we were testing the drug in animal model. So, phase 1 is a first in human trial. Now, how many people are involved in phase 1 clinical trial? Around 20 to 80 people are involved in phase 1 clinical trial. Phase 1 trial is further divided into two parts: single ascending dose study and multiple ascending dose study. So I'm not going into detail about what is ascending dose study and multiple ascending dose study now, considering the time. But I'll tell you what we do exactly in phase 1 clinical trial. So in phase 1 clinical trial, which is first in human trial, we determine the safety and maximum tolerated dose and PK parameter. Now, what is safety? As I said, safety means subject is not having unintended effect of the drug. For example, if drug is meant for blood pressure, it should not cause constipation, it should not cause diarrhea, it should not cause malaise, or it should not cause high blood pressure, it should not cause diabetes. So, unintended effect of the drug. Okay, those effects should not be there. So, by measuring those effects, we'll see whether drug is safe or not. PK parameter. Now, PK parameter means the absorption, distribution, metabolism, and excretion. Okay. Then third thing is maximum tolerated dose. Okay, this is very important, and this is the key thing about the phase 1 trial: maximum tolerated dose. So, maximum tolerated dose we'll understand with the help of one example. For example, three people you have recruited in phase 1 clinical trial, and you have given 50 mg of drug. Okay, and then you have increased the amount of drug, like for another three subjects you have given 100 mg. Now in these three subjects, one subject is having some unacceptable toxicity. Okay, suppose he is having high blood pressure because of your drug at 100 mg. Now with this we can again we will recruit some more subjects, say three subjects we have recruited more. Again one subject is having the same toxicity. Now this 100 mg is our maximum tolerated dose. Means with the name itself you can understand, right? The dose which can be tolerated to the maximum extent by any human being, that will be maximum tolerated dose. That dose we will determine in phase 1. So the key takeaway from the phase 1 trial is it's a first in human trial first of all. And how many subjects are recruited in phase 1 clinical trial? Around 20 to 80. Okay. Then what are the things which we measure in phase 1 clinical trial? We measure the safety, we measure the PK parameter, and then we measure the maximum tolerated dose. Okay, MTD. Now one more thing. Generally, the phase 1 clinical trial is done in a healthy volunteers. Okay, because in phase 1 there are many reasons why we do the phase 1 clinical trial in healthy volunteer, but the main two reasons are: the first one is the healthy volunteer will be able to tolerate the drug, right, compared to the ill patients. And the second thing is, in healthy volunteer we will be able to establish the maximum tolerated dose, safety, and PK parameter better compared to ill patients. for ill patient, the problem is their kidney function may not be normal, their liver function may not be normal, they have some cardiac issue, they may have some comorbidities, right? It will interfere with our results. So it's better to establish the safety PK and maximum tolerated dose in healthy volunteer. But there are some exceptions as well. Like for example, if you are dealing with cancer drugs are very toxic in nature, right? So generally we do the cancer trials in cancer patient itself. Okay, in phase 1 trial, in if drug is meant for cancer subjects, then we will recruit the cancer patients only. We will not recruit the healthy volunteer in phase 1. Okay, but in general, healthy volunteers are recruited in phase 1. So coming to phase 2. After phase 1, if results are desirable, then the drug molecule will be tested in phase 2 trial. So what is phase 2 trial? Phase 2 trial is also called proof of concept trial, POC trial. Okay, they are non-pivotal trial. Non-pivotal means whenever the data of that particular trial is submitted to regulatory authority, that trial is called pivotal trial. But data of phase 2 trial is not submitted to regulatory authority for approval purpose. Means with the phase 2 trial data, you cannot get the approval of the drug. And it's a proof of concept trial. Means here we will check the efficacy of the drug, whether our drug is working or not. Whereas in phase 1, if you will remember, in phase 1, we have not checked the efficacy. In phase 1, we are not testing whether the drug is working or not. Here, our main concern is safety. Once safety is established in phase 2, we'll see whether drug is working or not, whether drug is efficacious or not. Efficacy means how well the drug is working. Okay. If it's an anti-BP drug, anti-blood pressure drug, then we will see how well the drug is able to reduce the blood pressure. If it is anti-cancer drug or anti-tumor drug, we will see how well the drug is able to reduce the tumor size. Okay, that is called efficacy. In phase 2, our main concern is to determine efficacy, and it is also called proof of concept trial, and around 100 to 300 people are recruited. In phase 2 trial, we have to recruit the diseased patient, the patient which is having actual disease for which we are developing the drug. If we are developing the drug for blood pressure people, the person who is suffering from high blood pressure, in that case, we have to recruit the people with BP, high BP only in phase 2. Whereas in phase 1, generally we recruit healthy volunteers. Okay. Now again, and phase 2 is divided in two parts. So we will discuss the further division of phase 2 part in our detailed classes. Now I'm going to phase 3 now. So in phase 2, what we have determined in phase 2, we have determined the efficacy, how well the drug is working. In phase 1, we have determined the safety. Okay. In phase 3, we will further confirm. What we will further confirm? We will further confirm the safety and effectiveness. See, efficacy and effectiveness words are same. Whenever you are dealing with large number of people, then we will generally we will use the word effectiveness. We do not use the word efficacy. In phase 2 trial, we are dealing with only 300 people, right? So we are using the word efficacy. Whereas in phase 3, we deals with number of people, close to 3,000, sometimes 5,000 and 10,000 as well. So we are dealing with large number of people. That's why we are calling we are using the effectiveness word instead of efficacy word. And we will also confirm the safety part as well. So in phase 3 is a further confirmatory trial where we do the final confirmation on safety and effectiveness part. Okay. In phase 3 part, as it is done in large number of people, in thousands of people. So we can determine the drug-drug interaction as well. For example, we are doing the research on blood pressure lowering drug, the drug which lower the blood pressure. But person might be having the diabetes and he might be taking the diabetic drug. So in phase 3 trial, we will determine whether our drug is interacting with diabetic drug or not. So we can rule out the possibility of drug-drug interaction as well. We can also rule out the possibility of drug-food interaction as well. For example, some drug like tetracycline, if you will take with milk, the tetracycline can make a bond with calcium present in the milk. So those kind of interaction we can check. If we are dealing with the antidepressant drug, the drug which is used in the depression uh, disease, in that case it may have some interactions with uh, some other drugs, okay, some other food, okay. uh, so these kind of things, drug interaction, drug food interactions, and other side effect, and the further confirmation of safety and effectiveness takes place in phase 3 clinical trial, which is done in large number of people, like close to 1000, 2000, 3000, or up to 10,000. And it is also called pivotal trial or therapeutic confirmatory trial. So with the name you can understand, right? It is therapeutic confirmatory trial because it's giving the confirmation about the safety and effectiveness of the drug molecule. And it's a pivotal trial. Why it is a pivotal trial? Because the phase 3 data, the data from the phase 3 clinical trial is submitted to the regulatory authority for approval purpose. Phase 1 trial data, though phase 1 trial data is also submitted for the regulatory authority, but it is not submitted for approval purpose. Based on phase 1 data, regulatory authority, whether it is CDSCO or FDA, cannot approve the drug. Based on phase 2 data, regulatory authority cannot approve the drug. But based on phase 3 data, if it is acceptable, the drug is effective, safe, it's under safety parameters are well controlled, then regulatory authority can approve the drug based on phase 3 data. Okay. Now, again, phase 3 is further divided into phase 3 and phase 3B. So I'm not going into much detail about it. So now coming to the phase 4. Now, after the phase 3 data, drug is launched in the market. Okay. A drug is launched in the market. Now, in clinical trial setting, things are well controlled. Okay. Subject is coming to the hospital, pharmacist is giving the drug, in front of nurse only the person is taking the drug. We have well control about the circumstances as well. We are only dealing with certain people with certain diseases. For example, we are doing research in India, in Punjab region. Then it's possible that only the people who are living near Punjab will come to the clinical trials, right? So in clinical trial things are very restrict and well controlled, but in real life things are not well controlled. Person may have hypertension, person may have diabetes, the person's kidney function may not be normal, liver function may not be normal. Okay, he might be suffering from depression. he his food habit might be different. He would be living in a hill area. He would be taking some different kind of food. So in real world things are totally different compared to the clinical setting. So again we have to take care about the safety part in a real world scenario. So to check the drug performance in real world scenario is done in phase 4. That is called post marketing surveillance studies. Okay. So when it is done when once the drug is approved. So what do we do in phase 4? To know the long term risk and benefit of using drug, and to know the rare side effect, because in clinical setting only 3000 5000 people are there. Okay, rare side effect occurs in 1 in 10 lakh people, 1 in 1 crore people. So those kind of rare side effect cannot be measured in clinical trial setting. So those kind of rare side effect comes into picture or become visible when drug is given to the large number of people. Okay, I'll give you one example. There is one drug, roficoxib. It's a painkiller drug. Okay, it was withdrawn from the market due to cardiovascular toxicity. Okay, this toxicity was not observed in a clinical trial setting. When drug was launched in the market, the cardiovascular toxicity, people were getting heart failure because of consumption of this drug. So it was observed in phase 4 clinical trial. So you can understand why phase 4 is very important. Now we have learned phase 1 trial, we have learned discovery, preclinical, then phase 1, then phase 2, then phase 3, then phase 4. There is one more study that's called phase 0 trial. It's relatively new concept. Okay, it's not done with every trial. It's not done with every drug. It is done for few drugs only. So it's a relatively a new concept, and it's called microdosing studies or phase 2 trial, and it is done in very less number of people. It is done like in 5-6 people or max to max 10 people. So it is done before phase 1 trial, and drug is administered at subtherapeutic dose. Means what is subtherapeutic dose? Means the drug, the quantity of drug should be such that it does not produce any therapeutic effect in the body. Okay, so why we do? By administering this particular drug in a minute quantity, we can determine the ADME, absorption, distribution, metabolism, and excretion in a human body. Because if you remember, in after preclinical stage of development, we start the phase 1. What in preclinical stage of development, we determine the ADME, PK parameters, absorption, distribution, metabolism, and excretion in animal models. So though animal models give the suggestion like how drug is going to behave in human body, but our human organs and animals organs are different, right? Their liver function is different, their kidney function is different. They are metabolizing the drug in a different rate. Their biochemistry is different. So to have the good idea about the ADME, absorption, distribution, metabolism, and excretion, we give this drug in a subtherapeutic dose to the human. Okay, and we determine the ADME. So it's an empirical approach, means the practical approach to determine or to predict the human PK parameters, ADME. And by conducting phase 0 trial, the sponsor companies or the pharmaceutical companies can evaluate properly whether we have to go ahead with that particular drug or not. Okay. Yeah, that's all from my side. If you have any question, please ask. Anybody is having any question?
Okay. Okay, if no questions, then second topics, the pharmacovigilance will be taken by Ananthi ma'am. So Ananthi ma'am, you can share your screen. Meantime, you will receive one form on your chat box. You can fill the details so that the certifications of webinar will be provided to you. Okay, so just fill out the form.
Am I audible?
Meantime, yes, you are audible, ma'am.
Okay.
Yes, Sachin, you'll find a link actually. On this link, you have to be share your details. Okay, yeah, you can see the link here. Madhuri madam has provided some form. Fill this form only. You'll get the certifications. Okay. Yes, madam, you can go ahead.
Okay. Shall I start?
Yes, you can go ahead.
Okay. So, hi everyone. Good afternoon. Okay. So my name is Anandi. Okay, I'm going to be a pharmacovigilance trainer. So yeah, I have overall 10-year experience in pharmacovigilance. Okay, I work with Accenture and IQVIA, and here I'm doing this training sector as a part-time job. So last 7 years I'm doing this, training the candidates in job-based code. Okay, this is about myself. And today I'm going to discuss you about the basics of pharmacovigilance and what you are going to learn, what's the importance of pharmacovigilance, and what is the scope and what are the database we are using. So about that, the brief outline I'm going to give today, right? So what is pharmacovigilance? Any idea? So I hope all are health science graduates, right? So what do you mean by pharmacovigilance? So what is the importance? Anyone having an idea about pharmacovigilance?
Yes.
Okay.
Detection and assessment of the adverse drug reaction.
Yes, detection and yes, assessment of the adverse drug reactions. Yeah, it's perfectly correct. Okay, so here, so you know, right? Currently maximum we are using is allopathy drug, right? So none of the allopathy drug is 100% safe. Okay, each drug having some toxic effect. Okay, the minimal risk and maximum benefit going to enter into the market. Okay, none of the allopathy drug is 100% safe. Okay, so that is what you can see here. So medicine safety. So to undergo a treatment, you have to be very healthy because apart from your sickness, you have to withstand the medicine's effect. So what is the meaning? So the allopathy drug going to give a faster cure, but it having harm also, the toxic effect also. Okay. So that is why they have given like this. Okay. None of the allopathy drug is 100% safe. That is main. Okay. So here, so what are the correct things and what are the wrong things about the risk benefit? We are going to see now. So risk benefit balance of medications. The first one, medicines are safe. No, all the medicines are not 100% safe. Okay. So approved medicines are safe. So phase 0 done, 1 done, phase 2, phase 3, phase 4, everything is done. So it is safe? No, because in phase 0, phase 1, phase 2, phase 3, everywhere, little by little, we are understanding the drug's effect on the human body. Okay, even after phase 4 also, some drug is going to show the toxic effect after entering into the market. Okay, so we can't say that all the approved drugs are safe. Okay, meanwhile, we can't say that no medication is safe actually, because only the medicine is safe on human, only the drug is entering into the market. Okay, so it is not like all the drugs are not safe, but having harm also. Based upon this analysis only, the government going to decide that the drug is going to the market or not. Okay, so what is going to be a correct sentence then? No medicine is without risk. Yes, so none of the allopathy drug is without risk. It is having a risk, but when the risk is minimum or the max benefit is maximum, the drug is entering into the market. Right? See here. Right? So risk benefit balance of medicine. So when the benefit is higher, the weightage of benefit is high and risk is minimum, the drug is acceptable to the market. Okay. Next, so when the benefit is less and risk is going to be very heavier, so that time the drug is unacceptable. Okay, if it's a clinical trial drug and we are finding out the drug is having a higher risk, that time the trial is terminated during a phase 0 or 1 or 2 or 3 or phase 4. The trial is going to get terminated at any point of time. The drug is going to give some serious adverse events. Okay, so when some point of time the drug is going to give a serious adverse event after entering into the market. When after entering into the market, that time the drug is banned from the market, because some drug will show toxic effect immediately, some drug in continuous use, some drugs in prolonged use. Okay, like when the drug is going to give a toxic effect immediately, it's going to terminate during a research itself. But some drugs, you can say, take some common example as a diabetic drug. Okay, so all the diabetic patients going to take for long time, right? Their full lifelong they have to take. For example, starting at the age of 40 or 50, they want to take 60, 70, 80 till when they are going to live their life. It is 99% they're going to continue the drug, right? So in that type of scenario, we can't do a research for 40 years and we can find out all those things, right? So the research is going to happen approximately for 12 to 15 years. Okay, in that time we can able to find out some important side effects that is occurring within the 10 years of time, right? Not more than that. But the diabetic drug, right? So after 40 years of use, surely it's going to affect the kidney, right? You can, because it having a lot of residues, the allopathy drugs are having a lot of residues, so it's going to affect the kidney. A lot of kidney issues may occur for the diabetic patient. Okay, it's a prolonged use, long-term use toxicity that we can find out during a research. It can be find out only the drug is taken for long time. So that is why pharmacovigilance we are going to do for the clinical trial drugs as well as for the marketed drugs. Okay, so clinical trial drugs, if you're going to find out the toxic effect, then the drug is banned from the market. When the toxic effect is find out for the marketed drug, that time the drug is banned from the market. So termination, banning going to happen based upon the risk benefit analysis. That is why in pharmacovigilance, risk benefit analysis is very, very important. Okay, when the drug is giving some non-serious adverse event, that time the drug can continue in the market, no problem. Okay, when the drug is giving serious adverse event, so what does mean by serious adverse event? So the drug is going to give death. Okay, because of the toxic effect of the drug, patient died. Okay, or patient having some life-threatening condition because of the drug, the patient getting some life-threatening conditions, or patient hospitalized more than 24 hours in the hospital, or patient had any permanent disability in the body, or any congenital deformity, birth defect. Okay, pregnant lady taken the drug and baby got some deformities. So like that anything occurred. Okay, or anything comes under the EMA list, European Medical Agency's list, that is a seriousness list is there. So any out of which any one going to be there, the seriousness code, then it's a serious adverse event. Okay, any serious adverse event which is reported, then the drug is going to get banned. Okay, so how we are going to finalize? Okay, the drug is the reason for the serious adverse event. That is work of pharmacovigilance. So patient taken the drug, got some adverse event, and our main activity is whether the reported adverse event is occurred because of the drug or because of any comorbid condition, because of drug-drug interaction, because of drug-food interaction. So what is the reason for the toxic effect? Analyzing and confirming, yes, the drug is the reason for the adverse event. No, the drug is not the reason for the adverse event. So the causality assessment, it is called as. Okay, so analyzing the relationship and reporting to the government, it is work of pharmacovigilance. Okay. The definition. So pharmacovigilance is a science and activities relating to the detection, assessment, understanding and prevention of adverse effect or any other medication related problems. So first we want to get the report. We want to assess it whether the drug and the adverse in having a relationship. After detecting it, we want to report that to the government. Okay, why we are doing it? So prevent the adverse effect in future. That is the main motto of this pharmacovigilance. You can see here. So we are going to assess and understand the basics of the adverse event classification. You are going to detect it, whether what is the reason, and you are going to prevent the adverse event. For future occurrence, you are going to prevent it. So for the what we are going to do, we are going to do in the pharmacovigilance. Okay, so this is a very, very core criteria why the pharmacovigilance boomed up. Okay, the thalidomide effect, very, very important effect. It has been actually, the drug has been designed and entered the market in 1957. Okay, the drug is commonly used for morning sickness for pregnant ladies, right? The first 3 months they'll be having vomiting, dizziness, very severe, right? It's going to be. So that time the drug is used. Thalidomide drug is used for morning sickness. So very effective drug. So whomever the mother taken that drug in the first trimester, they feel very better on the morning sickness. But what is the harmful side effect is, it is going to give phocomela. Actually, what is phocomela? So whomever the mother taken the drug thalidomide, all the baby born without limbs. You can see in the picture, right? So no hands, no limbs, without limbs the baby born. Okay, then only everybody got very stunned. Okay, the single drug can able to give this many severe side effects. Actually, during the research, no one found out. So during the research, no one found out that there is a serious adverse event. But after entering into the market, they found out. Okay, the drug has entered into the market in 1957, and in 1959 only they have found out the serious adverse event of the drug. Okay, so 13 reports has been given about the birth defect in 1961, but overall more than 10,000 infants has been affected because of the thalidomide effect. Okay, so that this is term is phocomela, that deformity term is phocomela. So phocomela, it means the baby born without limbs. Okay, no hands, no legs, without limbs, the baby born. So this is called as keratogenicity. What is keratogenicity? The birth defect is called as keratogenicity. The drug is going to give some adverse event to the fetus. So the fetus is going to get some any birth defect because of the drug's side effects or its toxic effect. Then that is called as keratogenicity. So that is why in clinical trial also on animal studies we are conducting this keratogenicity study also. It's a very important motto of animal studies. We want to find the toxic effect of the drugs on animals. Okay, so that's why in clinical trial, let's say we are going to induce the drug to the pregnant animals to see whether the fetus is getting affected or not. Okay, because for the betterment of human safety, right? Because directly we can't enroll pregnant ladies in clinical trial. We can't endanger their life of the fetus, right? So in animal itself, we are going to do all this type of research and finding out the drug is safe. Then only is entering to the clinical trial on humans. In human study also, we are going to analyze actually the pregnant ladies and kids are going to be a vulnerable participant. Okay, they are having maximum risk. So maximum we will not allow those type of vulnerable participant into the clinical trial. In very rare scenario only we are going to use that. Okay, so this is one of the best example when the pharmacovigilance boomed up. Okay, because of thalidomide single drug, these big serious adverse event going to come in the sense of what is going to be the other drugs question mark, right? So safety of the drugs. So that's why they have given a lot of importance for the safety profile of the drug. So thalidomide disaster is one of the important milestone of pharmacovigilance when the WHO has started a lot of drug safety programs all over the world. Okay. Yes. The reasons for why pharmacovigilance is required. There is few reasons I'm going to tell you now. So first one is human consent. Okay. So as I told you, we can't able to find out all the safety of the drugs during research itself. Phase 0, phase 1, phase 2, phase 3, phase 4. We can't find out all the drugs side effects, that is adverse events. Okay. So that is why we are going to keep on continue in the clinical trial as well as marketing also. So phase 0, phase 1, phase 2, phase 3, phase 4, pharmacovigilance will be happening. Okay, whatever adverse event is reported to the clinical trial, that should be reported to the pharmacovigilance department within 24 hours. Serious adverse event. If it is non-serious, within 7 days we can submit. But if it is serious adverse event is reported, we have to report within 24 hours to the PV department. Okay, so we are going to create the case for the patient. You are going to process. You are going to do medical coding. You are going to do narrative writing, causality assessment, and you are going to report that to the government. Okay, so it is main that because for the humanity, because if anyone going to get the adverse event, it should be reported. Okay, actually in India we don't have that much awareness, but in other countries they are having a lot of awareness that the pharmacovigilance reporting programs are there. But in India it is booming up actually, because now in India, right, we are doing research for maximum of the other countries projects only. So US, UK, Australia, different countries, pharmaceutical companies only, we are doing pharmacovigilance activities. In India, in Indian patients adverse event, maximum government are taking care. The government are doing a lot of pilot projects all over, and they're having a lot of pharmacovigilance program like national PV program and zonal PV program and state PV program. We have different type of pharmacovigilance programs conducted by the government, and it's going on. But in India, we are working on MNCs for maximum of the other countries adverse event reporting only, not for our same own countries people. Yes, so it's already discussed by the previous trainer about the clinical trial. So you can see in phase 0 we are using very minimal participant. We are going to see about 20 to 50 participants. Why we are using healthy volunteers in phase 1? Because to see about the pharmacological activity, you don't need a diseased volunteer. Okay, so you don't want to disturb in the initial stages. So that's why you're using only healthy volunteer to see about the pharmacokinetics and dynamics of the drug. Okay, that time we'll see about the what is the basic toxicity, what is the pharmacokinetic action, ADME action, absorption, distribution, metabolism, elimination action, and everything we're going to see in the basic things. About the pharmacology, complete pharmacological thing we're going to see in the phase 1. And phase 2, we are going to use a diseased patient, because in phase 2, we want to find out the efficacy of the drug. So for that, you want a diseased patient. Okay, we're going to use a diseased patient in phase 2, and we're going to analyze how the drug is working on a specific disease and what's the optimum dose you're going to find out. In phase 3, you're going to use around 3,000 and plus participant. Okay, so here we are going to see about the different outcomes of the safety. Here, because in phase 3 only you're going to use lot of comparators. Okay, Like you're going to use like a standard drug, placebos, and different dosage regimens of investigational drugs, a different type of comparative trial you're going to do in the phase 3. And here you're going to find out the maximum of the safety profile of the drug also. Okay, so you're going to prove the drug is not superior to the other drugs and not inferior to the other drugs or equal to the other drugs. The comparisons are going to prove here. So here we can find out a lot of safety information in phase 3. So after all this, the drug is going for the NDA approval, new drug application. Okay. So when the NDA going to give you give approval for your phase 4, when in phase 3, the risk benefit analysis I have shown you, right? The risk benefit analysis. So in that the risk is lower and the benefit is higher, like that the proven data is there till the phase 3. Then only that NDA going to give approval for the phase 4, post-marketing surveillance. So in phase 4, the drug entering into the market in real life behavior, you're going to analyze how much the drug is safe in the real life behavior. You're going to analyze it. So all this safety data, pharmacovigilance is going to capture and report to the government separately. In clinical trials, they will be reporting separately through the clinical database. In pharmacovigilance, we are going to separately report to the government the same safety profile through the safety database like Argus, RSG, different softwares we are using. So through the software, we are going to report the safety profile from phase 0 to phase 4 to the government separately. Okay. So based mainly who's a regulatory authority, the government bodies. The government bodies who having authority to approve or deny the market authorization for the drug. Okay. Each country having their own regulatory authorities. So like who's our country authority? CDSCO, Central Drug Standard Control Organization. Like that we having US FDA, Europe's EMEA, each country having their own regulatory authority. So in which country you are going to do a protocol, according to that you want to prepare the documentation, rules and regulations, everything is going to be different, right? So in phase 1, phase 2, phase 3, phase 4, everywhere we want to get approval from our government, which country protocol it is. So according to that you want to get the government approval. Okay. So after phase 4, the drug is entering into the market. After entering also, the drug can give some serious adverse event. That is why there is no end for pharmacovigilance. So from phase 0 to phase 4, after marketing, till the last human taking the drug and the drug is banned from the market, till then the pharmacovigilance activity is going to get continued. There is no end for pharmacovigilance, right? So reason 2, okay, medicines are supposed to be save life. Okay, so dying from a disease is something unavoidable. Okay, so if the patient got the disease and we treated, patient died, that is acceptable. But dying for the medicines is unacceptable. Okay, because the drug is having a serious adverse event. Just because of the serious adverse event, patient died because of the serious adverse event, it is not acceptable. That time only the drug is banned from the market. That is why pharmacovigilance we are doing. Second reason for doing pharmacovigilance, to save the human life. Okay. So in US and UK, right, maximum of the common death cause is going to be the adverse drug reaction. That is in US, it is 4th and 6th commonest cause. Okay, in UK, more than 5,700 deaths happen per year in UK. So what is a, so that is why pharmacovigilance is boomed up. US, right, they are 100% concentrating on the pharmacovigilance activities because as the commonest cause of 4th and 6th place goes to this ADR, right? So that is why they have collaborated with the WHO, ICH. They have collaborated and they have created a lot of pharmacovigilance program. So 3 countries are called as ICH countries: Japan, US, and Europe. So they only started initially creating these ICH guidelines as well as that pharmacovigilance activities, everything they have started. Now all the countries has been enrolled in that, right? Third reason, ADRs are expensive than the drugs research, it seems, because if any of the approved drug in the market are given the serious adverse event, that in the patient can put a legal case on the company, right? The company want to spend million of amount as a compensation to the patient. So more than doing a research, right, for the mortality rate, because of that compensation giving and because of the adverse event, lot of expenses are happening for the government also to treat the patients. So because of that, the third reason, the drug is giving a maximum of the expense. Okay, that is the main reason where they are, the pharmaceutical companies are giving more importance for pharmacovigilance, because if any serious adverse event occur from their side, they want to give compensation millions, right? And fourth reason, promoting rational use of the medicine. So exactly to treat what disease, to what amount of drug should be taken, in what routine, in what duration. So everything should be pakka. It should be very clear. Patient should know everything. Okay. So one time the doctor has prescribed some drug, right? What the patient will do? Next time they are not feeling well for the same type of indication, they will use a self-medication. Self-medication is very, very dangerous. We should not take self-medication. So OTC drugs are accepted. Paracetamol, common drug. So for fever, for body pain, yes, it is accepted. But for some serious events, please don't go for self-medication because it's going to cause a very serious adverse event. Okay, in prolonged use or a wrong time you are taking a drug, or for a wrong indication you are taking a drug, it's going to give you a serious adverse event. That is why rational use. You want to find out exactly for what you want to treat. That is reason 4. And reason 5, ensuring public confidence. Okay, a patient should have a confidence on the drug. Okay, if I'm going to take a drug, I'm in a safer hand. I'm going to get treated for my disease and I'm going to be in a safer hand. That confidence the patient should get. That is 5th reason. And 6th one, ethics. There's a moral value. Okay, so how the human should be treated because of the drug. So ethics to know of something that it is harm to another person who doesn't know about it. We want to tell them. So for that, what we want to do, we want to report the adverse event. Okay, I have taken the drug and I got the adverse event. Then I want to report that to the government. Then only other people going to get saved from the adverse event, right? So all these things I should think and I should report. That is why the government, they have given a lot of awareness, and many people started reporting through the government website. It's they are having, each and every country having their own government adverse event reporting websites. They are doing that, and it is coming to the pharmaceutical company, and pharmaceutical company forwarding to the PV department. So you are going to work for the pharmaceutical company's PV department. Okay, so these are the main reason why you want to do the pharmacovigilance program. Okay, so because of difference in drug production. So why we want to do pharmacovigilance in every country? Because there is a difference in drug production, distribution of use. That is for indication, different dose, different availability of drugs, and different genetic variation. So the each and every human having a different genetic variation, right? So Indians are different, US person different, Europes are different. The genetic variations will be there. So because of the genetic variation, drug effect also will be different. So because of diet, there will be a interaction will be there. Because of traditional medicines we are going to use, or because of the interaction, any adverse event we may get. Lot of chances are there. And because of the pharmaceutical products quality. Okay, so we are using excipients like coloring agent, binding agent, flavoring agent with the active ingredient, right? So same active ingredient, but different company product going to give you different adverse event, because the active ingredient is going to be safe only, but the other ingredients like excipients we are using, right, that also can cause some serious adverse event. That is why we are going to keep on doing pharmacovigilance for all the drugs. Okay, so that is why the WHO's program has been initiated. Now all the country has been joined hands with WHO, and all are doing the pharmacovigilance program. The governments are doing the pharmacovigilance program. The zonal, regional, national level pharmacovigilance programs are happening. So zonal means in each and every states that government hospitals are there, right? So there they are going to collect the adverse event report from the inpatients, and that is going to transfer to the regional zone, and from regional it is transferred to the national zone, and from national pharmacovigilance center, that serious adverse events that having a relationship with the drug that is going to shared with the WHO. In the World Medical Association, in WHO, it is going to be shared. And so we discussed worldwide, and they're going to decide what they can do to the improvement of safety of the patient. So this is what the overall pharmacovigilance activity. Okay, now I hope everyone got what is pharmacovigilance and why we are doing it, what is the importance. Let's come into the live process. Okay, this is what pharmacovigilance is, and the live process is the database. Okay, so in where you're going to work means, as a pharmacovigilance associate, you're going to work in MNC like Accenture, Cognizant, different companies you're going to work. What is your role? You're going to work in the database, completely system-based job. Pharmacovigilance is a system-based job. You're going to work on the system. So what you're going to do? So here you're going to have it. You're going to use two databases, Argus, RSG. So it's also called as Aware and Shaffer. Okay, these databases only commonly used in India for do the pharmacovigilance activity, safety reporting. Okay, so pharmacovigilance database is also called as safety database.
Hello.
It's also called as safety database.
Ma'am,
Okay.
can you explain in detail? Which database is used?
Argus, ARGUS, Argus database, ARIS, ARIS, Argus database, ARIS database. Two database you're going to learn from my classes. Okay, two databases, Argus and ARIS. Both are same content only you're going to enter, but the alignment of the database is going to be different. Argus is a very important database. All over the India we are using. More than 80% Argus only we are using. ARIS 20% we are using. Argus we are using more than 80%. Right? So in the Argus database, you are going to learn in the classes. So what is pharmacovigilance? What are different type of pharmacovigilance? Because pharmacovigilance is not a single type. We are having ICSR, that is individual case safety report. Each and every individual patient case you are going to process and report. That is one type of pharmacovigilance. For example, we are in pharmacovigilance department. Okay, we are getting 500 cases. So out of 500 cases, you are going to prepare 500 different cases. We are going to create for each and every 500 patient. One by one patient, you are going to process. You are going to create one case. You are going to process the case. You are going to analyze the case, and you are going to report to the government. Like each and every patient case, processing and reporting is single case processing. It is one type of pharmacovigilance. Another type is aggregate reporting. So aggregate reporting is another type of pharmacovigilance. Here you are going to collect the drug information for prolonged period of time. Okay, single, for example, take a common drug paracetamol. Okay, for a single drug paracetamol, you are going to get some 10 cases per day, for example. So per month, how many you will get? 300 you will get. So per year, how many you will get? More than 3,000 you will get, right? So I am going to collect the drug paracetamol's adverse event for prolonged period of time, 6 months to 1 year. So what I will get? I will get more than 3,000 to 5,000 cases about the paracetamol drug. So what I'm going to get? I'm going to get a broad review about the drug paracetamol. Okay, so I'm going to get the safety profile of the drug by getting the single drug information for prolonged period of time, and that is going to be reported like a what is that? Literature. Like that, you're going to prepare a documentation, the safety profile that is going to be shared with the government. That is aggregate reporting. Each and every case reporting is single case reporting. Collecting the drug for prolonged period of time, and that is going to be reported is called as aggregate reporting. So about single case reporting, about aggregate reporting, you're going to see in brief in the classes. Okay, and database training, Argus, RSG database training going to happen. So exactly in workplace, what database you are going to work, I'm going to show that slides to you. I'm going to explain you how you're going to work on the database. Okay, so you're going to get the 100% workflow knowledge. Okay, in MNC, how you're going to work, same knowledge you will gain in from my classes. Okay, each and every database, please, I will explain you what is the mandatory fields, how you want to enter, how you want to write the narrative writing, how you want to do medical coding, how you want to report to the government. Everything I am going to explain you in the classes about pharmacovigilance. So in the classes, you are going to learn about the core of pharmacovigilance, different types of pharmacovigilance, database, entry-based questions. So all this is going to be covered in my classes on pharmacovigilance. Okay, so this is a outline of pharmacovigilance. For demonstration, I think it will be clear for you. So any doubts you have in pharmacovigilance, you can ask me now. Anyone having a doubt in pharmacovigilance? Feel free to ask me. Okay, whatever you're having doubts in PV, you can ask me.
I think nobody has any questions. So let me share my screen to detail out about the Company Connect Consultancies actually. So Company Connect Consultancy providing the various training actually related to pharmaceutical industry, related to IT industries and clinical research industries as well. Okay. So this training program is related to pharmacovigilance, clinical research, clinical data management, and medical device industry, medical device programs actually. Okay, just give me a second. It includes the means the Company Connect provide the training on the computer system validations, pharmaceutical quality assurance, quality controls, pharmacovigilance and clinical research, and CDMS and medical writings. The equipment qualifications, pharmaceutical equipment qualification, HVAC validations, cleaning validations, water system validation, and process validations. Then regulatory affairs courses. This is also courses we have. LIMS application, if someone having the knowledge on the applications. So LIMS application end to end, we can say course is there. You can enroll these courses. Under this, we have like a various training program. So one is the pharmacovigilance, clinical research, CDMS, and medical writing combo project actually. So that is the combo we can say program we have. Apart from that, you can enroll pharmacovigilance plus clinical research, our clinical data management system plus clinical research, and medical writing plus clinical research. Any of the program you can enroll actually. Okay. So currently you can also enroll for the combo pack so that it will be benefited to you. You can opt or apply for any of the vacancy that is in CDMS or clinical research or pharmacovigilance or medical writing actually. That will be very helpful for you actually. Career related things. So by going through these courses after completion of the Am I audible? Hello?
Hi, yes, now audible.
Okay. Yeah, thanks. So after completion of courses, you will be able to go in a clinical trial industry, in a consultancy industry like a TCS, Infosys, Mindtree, IQA. These are the consultancy industry who provide the consultancy into various CROs actually. And as well as you can go to the pharmaceutical industry in the field of the pharmacovigilance as well. The starting package of as a freshers will be 20,000 to 40,000 in India. Outside India, it will be much more actually. Relative career growth is 50% higher than any other courses, exposure to corporate environment, and you can enjoy the 5 days working actually. This is the like a career growth from the starting from the 2.16 to 5 lakhs per annum to goes beyond the 25 to 75 lakhs per annum with the these kind of a courses. Benefit to join our institute is like a placement assistance. So after completion of the course, a placement assistance will be provided to you. Access to study material, you can access the study material will be provided to you. You can download those study materials. You can also like join 2 or more batches with the same fees only. Means once you apply for registration, pay the payment, then you can join any of the batches actually. Means like if you did not understand in one batch, you can join the second batch as well. Recording the classes will be available on the site to recall and create your own notes actually. You will allow for the member chat area. So member chat area will be available so that you can discuss with the company students, like previous students as well as the current student. And you can also raise your questions if you have any questions, even after completing the course. Means it will be like a lifetime support. Whenever you have questions, you can ask the questions in a chat box through the member chat area. Okay. These are the course details, and you can contact to us if you want to be enrolled for these courses actually on below numbers. You can go and visit the company site www.companiesconnect.com, or you can have a WhatsApp on 96916 Any questions? Let us know.
Sir, yes, may I have one question?
Yes, go ahead.
What is combo charge? What is charge for combo course?
Okay, under the combo chart course will include all the things like pharmacovigilance details, clinical research details, CDMS details means clinical data management systems, and medical writing. This is the combo pack actually. Means all the courses related to CRO, related to pharmacovigilance will be included in this particular course actually. Okay.
What is charge?
What is? Hello?
Fees. What about fees?
The fees structures and all the Madhuri ma'am will provide. You can contact on these particular numbers for the fees details and the brochures. Brochure will be sent to you on your WhatsApp number or on the mail ID. And just go through the topics and the fees details in that. Okay, sir.
Or there is a 100% placement guarantee or not?
No. 100% nobody is going to be provide the 100% guarantee. If somebody is saying 100% guarantee, he's telling lie. If there is no vacancy in the market, how will he'll provide the 100% placements actually? But we do the assistance for the placement so that you will get placed in a good organization. So wherever vacancy is available in the market, we'll provide those vacancy details to you. Okay, sir. Any other questions?
You will
So we'll
refer or
try.
not
Yes,
particular
sir.
organization. If any vacancy come, you will refer.
Yeah, definitely we'll refer you. Okay. We'll provide our preference to our students first. Means we'll generally search out into market for the good candidates actually. If we have already our good candidate in our organizations, we'll refer those candidates for sure. Okay.
Oh,
Any other questions? Anything?
Sir, interview
Yeah, go ahead.
interview preparation will provide or not before going to attend in some companies for interview purpose.
Yes, a mock interview will be provided to you, sir. Okay, after completion of the course, a mock interview will be scheduled so that you can understand how the interview will be taking place actually. Okay, so a mock interview, 1 or 2 classes will be there. Even we'll share some mock interview questions and answers to your mail ID. You can just go through those questions and answers. Okay. Any other questions? So we can say once you'll complete the course, you will be able to understand and you will be able to fight any of the interviews in the CRO, in the IT industries as well.
what is duration of course, sir?
The duration of course is 1.5 months, but it's not at like a time dependent. Until unless we'll not cover all the topics, we'll complete the course maybe 2 months or maybe beyond the 2 month even. Okay, sir. So we'll cover as much as topics what we mentioned in our brochures actually. Any other questions? Okay. Okay, then I think that's it for these webinars. Just contact to us on below numbers if you wanted to be join the courses actually, any of the courses. Thanks for joining. Yes, go ahead.
Sir, how can we get the certificate of this session?
You have filled the form, correct? There is a form you may have seen in the chat box. Just open your chat and click on the form, fill out the details, whatever you have actually, and just we can say fill this form. The certifications will be provided to you. Okay, you can visit our website as well. Under this, you can create the login. You can details out all the things like a members. You can see the members details. You can chat with us. There's an option for the chatting after you complete the logins and all. Okay, sir. So that is how you can see all the members we have actually currently. Okay, just visit our website companyconnect.com.
Sir, I have one question also.
Just go ahead.
Sir, if I get enrolled in this Connect consultancy for these courses, combo courses, so
Yes.
what are the for these classes, sir? It's regular or it will going to conduct alternative days?
No, it's a regular Monday to Friday, 9 to 10 p.m.
9 to 10 p.m. Okay.
Yes, 9 to 10 p.m. Suppose if you miss the class, a recorded session will be available on the website. On the particular login, you will provide the access to you actually. By that page, you can just go through the recorded sessions actually. Okay.
Okay. Thank you.
Any other questions? So just fill out the form after. Yeah. And is any other questions to anyone? Am I audible? Hello?
Yes, you're audible.
Okay. Are you able to access the chat box? Just fill out this form so that we can reach out to you for the certifications. Meanwhile, any other questions to anyone? Let me know. Does everybody fill this form? Just mention yes, and you can leave the meeting then.
No, sir, just wait, please.
Yeah, sure, no issue. Just mention in a chat box yes, and you can leave this particular webinar after that. Once you fill out the form, you can leave this webinar, not an issue. If you have any questions, just reach out to us on the WhatsApp or through the mail so that we can answer those questions.